So today we're going to talk about determining function of non-coding RNAs. So for the last several decades, there's been a central dogma: DNA encodes for RNA, and RNA encodes for proteins, and proteins do most, but not all, but most of everything. However, it's pretty clear that RNAs also serve an important functional role. For example, there's the well-studied classes of tRNAs, rRNAs, or spliceosomal RNAs. And in the last decade, there have been several new classes of RNAs identified with broad regulatory roles. These include micro RNAs, which can inhibit translation or transcription or even induce gene activation; large intergenic non-coding RNAs, or links, which can silence genes or function as natural antisense transcripts or as scaffolds for proteins; natural siRNAs, or small interfering RNAs; pRNAs, or small nucleolar RNAs. Today we're going to concentrate on two classes: microRNAs and large intergenic non-coding RNAs. These were first discovered in C. elegans by the Ambrose lab about 20 years ago. Lin4 was known as a regulator of larval development, and it was known that the gene product regulates Lin14 via its 3' untranslated region. It was a mysterious how it was interacting until the Ambrose and Ruvkin labs noticed that the Lin4 sequence was complementary to multiple sites in the Lin14 3' untranslated region. Subsequent work demonstrated that Lin4 physically bound to the 3' UTR of Lin14 and downregulated its translation, and Lin4 became the founding member of a class of genes that are now known as microRNAs. These tend to be small, 18 to 23 nucleotide regulatory RNAs present in genomes ranging from plants to humans. They comprise about 4% of human genes and regulate somewhere between 10 and a third of human gene products. They can regulate gene expression by binding to complementary sequences in their targets, and in metazoans they can inhibit translation. And so far they've been implicated in many processes, maybe most biological processes in mammals. Here's a slide on the biogenesis of microRNAs. We have them being transcribed off of a host gene here. Then they can form a pre-mRNA or similar form and be spliced, or be immediately processed by the Drosha and Pasha enzymes into a pre-miRNA. They're then exported from the nucleus by this RAN-GTP exportin 5 pathway, where they are recognized by their dicer molecule, and this 3' hairpin here is cleaved, forming the miRNA, and this is then incorporated to the miRNP complex. MicroRNAs degrade mRNAs in both plants and animals. They also inhibit protein translation in animals and plants, and sometimes alter chromatin structure and function by changing histone modifications and/or DNA methylation patterns. The first question: what effect would the disruption of exportin 5 likely have on microRNAs? Would it cause A, sequence mutations, B, nuclear accumulation, C, cytoplasmic accumulation, or D, all of the above? It would be B, nuclear accumulation. Exportin 5 is involved in exporting precursors to microRNAs, and blocking that step would cause them to accumulate in the nucleus. So let's talk about finding their functions. There have been several approaches that have been used successfully to find the functions of microRNAs. These include expression profiling. By relating when and where microRNAs are expressed, it can inform what they might be doing. There are pure computational approaches, where a lot has been learned about their targeting and many computational approaches have been developed. There are functional screening approaches, where you can synthesize or encode in plasmids microRNAs and screen for their functions. And there are integrative approaches that use some or all three of the above methods. So here's an example by finding things by expression. Here's a paper from the Croce lab: frequent deletions and downregulation of microRNA genes miR15 and miR16 at 13q14 in chronic lymphocytic leukemia. And here's a second example from Liu et al. PLOS Genetics, published this year, where miR93 promotes mesenchymal to epithelial translations in breast epithelial cells. And here you can see the various populations of the cells here are in the facts plot, and how the expression of miR93 is regulating the type of cell that and the EMT process in this particular cancer cell line. Many labs have done exhaustive experimental techniques to develop computational rules to predict microRNA targeting. So this is a paper, classic paper from Brennicke et al. In PLOS Biology in 2005, where various mutations are introduced into a target site in an mRNA, and then the mRNA is incubated with a micro RNA in a cell, and the phenotype is observed for each mutation. And in this particular work, they concluded that nucleotides 2 through 8 of the microRNA, now called the seed region, is critical for its activity. But there are versions on the theme. There is a 5' dominant seed region, which is a canonical microRNA targeting site seed. There is a 5' dominant seed model here in the miR4 condition here, and there's a 3' compensatory binding where additional homology at the 3' end can help in the recognition forming a stable complex that leads to target degradation. Question 2. A mutation in which of these nucleotides is likely to be most disruptive of a microRNA function? Is it nucleotide 5, 10, 15, or 20? We said that nucleotides 2 through 8 were the most important. And so it's answer A, nucleotide 5. Function by computation. Based on these experimental data, a number of groups have developed algorithms for microRNA target prediction, and these include PICTAR, Target Scan, RNA22, Microcosm, Diana MicroT, etc. These algorithms are primarily based on perfect match of seed regions, nucleotide 2 through 8 of the mRNA, with target transcripts, that the target site is located in the 3' untranslated region, that there's conservation of a microRNA target site across species, and that there can be 3' compensatory binding, where imperfect binding of a seed region is allowed considering there is sufficient complementarity in the 3' region. There's also a higher ranking for target sites closer to the ends of the 3' UTR, and favorable nucleotide composition of target sites are sometimes included in the above algorithms. Here's an example of functional screening, where you can use small molecules in a cell-based model and some readout. For example, a luminescence readout where high here would be depicted in red and low signal is depicted in green, and the intermediates are depicted in variations on that theme. And you can use RNAi or microRNA mimics to either knock out or overexpress microRNAs to sort of determine what they might be doing in a cellular model. So here's a design of an NF-kappa-B microRNA screen where there's a kappa-B element driving firefly luciferase expression, and that's integrated in a HEC293 T cell. And you transfect in a reporter cell line with a library, in this case, of 328 microRNA mimics. Wait 72 hours to allow the microRNAs to recognize their targets and for the effect on their targets to occur. And then you can, after some stimulation, measure luminescence and analyze the effect of the functional effect of these microRNAs. Question number 3. MicroRNA X is predicted to target a component of an important pathway. Why doesn't overexpression of microRNA X have a phenotype? It could be that A, microRNA X is already highly expressed and not limiting. B, the microRNA its predicted target aren't co-expressed. C, the predicted target isn't limiting in the pathway. D, just B and C, or E, A, B, and C. And of course the answer is A, B, and C. So here's an overview of the screen that I mentioned a couple of slides ago. Minus TNF, which is used to stimulate the NF-kappa-B pathway, or plus TNF. And here you can see that the log 10 luminescence values of all of the wells in the screen go up several fold by the addition of TNF. And here you can see a z-score plot where the screen has been repeated twice, once on this axis and once on this axis, and the performance of each microRNA is plotted. And if it was perfect, it would lie on a completely straight line here, which I don't seem to be able to draw, but a straight line here. And what you can see is that there's a very linear relationship between the two replicates, which means that it's not noise and more likely to give you a promising result. Here are some validations from the top hits from the NF-kappa B screen. Here's a negative control. Here are a few microRNAs that seem to have strong functional effects. MicroRNA 517A and C, which in the absence of TNF give a strong readout, and then 210 and 3D43P, which also induce signaling in the absence of TNF. So now you know that you have a few microRNAs that have a functional screen, but you don't exactly know how. So one strategy to find the causal targets might be to take the predicted targets of the microRNAs from the previous slide and then integrate them with functional data from a cDNA screen, looking for cDNAs that are known to repress the NF-kappa B pathway. And so when we did this, we found the target of the microRNAs 518A and C is TNEP1. So when you overexpress 517A or 517C, but not a closely related molecule 517B, you can see that TNEP1 protein levels go down. And over here, you can see that inhibition of TNEP1 using siRNAs, so inhibiting the message for TNEP1 directly, also has a strong stimulatory effect on the TNF pathway in the presence of IL-8 or TNF. True or false? There has to be a single causal target for a microRNA phenotype. That's false. Like protein encoding genes, microRNAs can target multiple steps in a pathway, and so even though the strategy I showed you was heavily reliant on a strong single edge, it may not always be the case that you have one strong single edge, and finding the targets of functional targets of microRNAs can be quite tricky. Right, let's switch gears and talk about links, or long non-coding RNAs. So for quite a while now, probably about a dozen years or so, we've known that there are quite a number of non-coding RNAs in the genome. So here are a couple early papers. One from the Riken group, where they cloned tens of thousands of cDNAs and sequenced them, and then looked for open reading frames in the sequence. And to their surprise, they found that a chunk, possibly 25%, did not have any discernible polypeptides in their sequences. And over here is a more recent paper from Mitch Gutman and Eric Lander and John Wren and colleagues, where they used chromatin signatures to find thousands of conserved large non-coding RNAs in the mammalian genome. So they're extremely prevalent. There are probably around 10,000 or so of these long non-coding RNAs, and they've also been identified by RNA expression dynamics, and this is largely work from Tom Gingeris's lab. But the big remaining question is, what are they doing? So we know that examples of them play roles in splicing or translation or in gene regulation, either directly via siRNA-mediated effects or by modifying chromatin structure and being involved in imprinting. And XIST, or the X-linked inactivation center transcript, is probably the most well-studied long non-coding RNA. So XIST inactivates one X chromosome. It's expressed from near the center of the X-linked inactivation center. It coats the inactive X, and it's negatively regulated itself by a non-coding RNA called T6. It's about 15 kilobases in mice, so these things can be quite large, and the knockout is early embryonic lethal in mice, and there is a disorder called familial skewed X inactivation, which occurs in humans. That's a mutation in this particular gene. So how do we find out what links do? The methods end up being very similar to to the methods we discussed earlier with microRNAs, save that there doesn't seem to be this easy to discern sequence signatures, unlike there are for microRNAs, with one exception, which are those links that are working as antisense. So here's a functional screening example from a paper from 2005, Willingham et al., where we used plasmid encoding shRNAs targeting 512 conserved links from between mouse and humans, and we screened them across a variety of cell-based of cellular pathways, including the NFAT pathway, and looked for activity-dependent hits. We found one long non-coder, which we renamed NRON, strongly activated the NFAT pathway in the absence of presence of stimulating molecules ionomycin and PMA. So here you can see 5 different shRNAs designed against NRON are giving you a phenotype, and we have at least 2. In this case, we have 3 shRNAs that are giving us a consistent phenotype. They're also doing it in a different cellular model, the JERCAT model. You can see that they're having a strong effect on the NFAT pathway, but not an effect on the NF-kappa B pathway, the P53 pathway, or the AP1 pathway. And we can see that by qPCR that the shRNAs are knocking down NRON in the same cells. After we chased down the mechanism, we looked for proteins that physically associated with. First, we actually looked at what happened to NFAT in the presence of NRON shRNAs, and we did this by using cellular imaging. And we noticed that when you put in shRNAs designed against NRON, NFAT became constitutively nuclear. So you can see very punctate nuclear signaling over here. And in contrast, if you had control shRNAs, NFAT remained cytosolic. And this is just a quantified version of the Western blots that are depicted down here, showing roughly the same thing, where we have NRON shRNAs tested against the FOXO1 reporter, or control shRNAs in the NFAT pathway, and then NRON shRNAs in the NFAT pathway, and those are having the strongest effect. Question number 5. The length of links causes what potential problems in finding their biochemical functions? Is it A, they're hard to clone? When are you sure you have the 5' or 3' ends? B, they form elaborate secondary structures? C, ATGs happen. There could be short peptides that are translated and functional. D, just A and C. Or E, A, B, and C. And the answer is D, just A and C. They may form elaborate secondary structures, but that doesn't really have a major impact on establishing their biochemical function. So how do you know that it's actually working as a non-coding RNA and that there isn't some cryptic translation and small peptide coming off of a long transcript? Remember that peptide hormones are also quite small. They can be only a few amino acids, but they also have major effects on biological pathways. Many work as natural antisense transcript, so we wanted to use a MS2 maltose binding protein fusion to determine the proteins that are interacting with NRON to figure out how it might be working on the NFAT pathway. And so when we did this, we found that NRON interacts with importance. The identities aren't really that important, although they're depicted here on this along this axis over here. And when we took these importance that NRON is physically interacting with and overexpressed them, over in this panel over here, you can see that IQ-GAP1, KPNB1, PPP2R1A, and PSMD11 are having relatively strong effects. And here you can see the percent expression by qPCR of all of these molecules in the lower panel. So nRON appears to be physically interacting with several components that are regulating the translocation of the NFAT protein as it goes from the cytoplasm to the nucleus during its signaling event. There are, of course, other classes of functional non-coding RNAs, endogenous siRNAs, Pb classes, and small nucleolar RNAs. So final thoughts. There are about 25,000 protein encoding genes in the human genome, but there are thousands more of ncRNAs. Are they genes? Many are not conserved. So are they things that distinguish primates from rodents? Or are they simply expressed sequences that don't have a real function? XIST is actually relatively poorly conserved between mouse and humans, but it's been demonstrated that it's absolutely necessary in both species. So we know the function of only a few dozen of these long non-coding RNAs, and there are thousands and thousands to go.